एवरीवन, वेलकम बैक टू माय यूट्यूब चैनल। आज की वीडियो में हम बात कर रहे हैं ओवरव्यू ऑफ lymphocyte एक्टिवेशन एंड अडेप्टिव इम्यून रिस्पांस। अडेप्टिव इम्यून रिस्पांस डेवलप इनटू स्टेप्स कंसिस्टिंग ऑफ द एंटीजन रिकॉग्निशन, एक्टिवेशन, proliferation and differentiation of specific lymphocyte into the effector and memory cell, elimination of the antigen and decline of the response, with memory cells being the long-lived survivor. The major events in each step are summarized next. These general principles apply to protective response against the microbes as well as pathological response that injure the host. Capture and display of antigen. Microbes and the other foreign antigens can enter the body virtually anywhere, and it's obviously impossible for the lymphocytes of the every specificity of the patrol every possible portal of the antigen entry. The overcome this problem, microbes and their protein antigen in epithelia and the other tissue are captured by resistant dendritic cells, which then carry their antigenic cargo to draining lymph nodes, through which the T cells constantly recirculate. Here the antigens are processed and displayed complex with the MHC molecules on the host cells, where the antigens are recognized by the T cells. Similarly, soluble antigens are captured and concentrated in the follicles in lymph nodes and the spleen, where they may be recognized by the B cell via their antigen receptor. All the same time as microbial antigens are recognized by the T lymphocyte and B lymphocyte, the microbes activate innate immune cells expressing the pattern recognition receptor. In the case of immunization with a protein antigen in the vaccine, microbial mimic called adjuvant and stimulate innate immune response given with the antigen. During the innate response, the microbe activate the APCs to express molecule called co-stimulator to secrete the cytokines that stimulate the proliferation and differentiation of the T lymphocyte. The principal co-stimulation for the T cells are B7 proteins, which expressed on the APCs and are recognized by the CD28 receptors to naive T cells. Antigen and co-stimulatory molecules produced during innate immune response to microbe signal to function cooperatively to activate the antigen specific lymphocyte requirement for the microbes trigger signal to ensure that the adaptive immune response is induced by the microbe and not by harmless substances. In immune response to tumors and transplant, signal 2 may be provided by the substance released from the necrotic cells. Damage associated molecular pattern mentioned earlier. The reaction and the functions of T lymphocyte and B lymphocyte differ in important ways and are best considered separately. Cell mediated immunity activation of the T lymphocyte and elimination of intracellular microbes. Naive T lymphocytes are activated by the antigen and co-stimulators in the peripheral lymphoid organ and proliferate and differentiate into the effector cell that migrate to inside where the antigen microbe is present. One of the earliest response of CD4 helper T cell is secretion of the cytokine IL-2 and expression of the high affinity receptor for interleukin 2. Interleukin 2 is a growth factor that affects these lymphocytes and stimulate their proliferation, leading to the increase in the number of the antigen-specific lymphocytes. The function of the helper T cells are mediated by the combined action of the CD40 ligand and cytokine. CD40 is a member of the TNF receptor family and CD40L is a membrane protein homologous to TNF, and when CD4 helper T cell recognize the antigen being displaced by the macrophages or by B lymphocyte, the T cell express the CD40L, which engage the CD40 on the macrophages or B cells and activate these cells. Some of these activated CD4 T cells differentiate into effector cells that secrete different sets of the cytokines and perform different functions. Cell of the Th1 subset secrete the cytokine IFN-mega, which is a potent macrophage activator. And the combination of CD40 IFN mediated activation result in the classical macrophages activation, leading to the induction of the microbial substance in the macrophages in the destruction of the ingested microbes. यहाँ तक कोई भी क्वेश्चन हो तो ज़रूर पूछ सकते हैं आप. नेक्स्ट वीडियो में इससे आगे का पार्ट डिस्कस करेंगे। तब तक के लिए इजाज़त चाहूँगी। अल्लाह हाफ़िज़।